The UPS marks a target destined for destruction by using a ubiquitous small protein found in all cells, aptly named ubiquitin. In essence, proteins tagged with ubiquitin are given a very visible death sentence. There's a lot of ubiquitin present in cells, but it can't attach itself to proteins at random. It is a highly regulated and controlled system to avoid any unwanted protein degradation. First, it must be primed for action by an E1 ubiquitin activating enzyme. This process also requires energy in the form of ATP, or adenosine triphosphate. The activated ubiquitin is then transferred from the E1 onto a second enzyme called an E2 ubiquitin conjugating enzyme. This enzyme acts as an escort for ubiquitin to its next destination, the E3 ligase enzyme. The E3 enzyme acts as a platform on which the target protein substrate and the active E2 ubiquitin complex can meet and interact. The E3 enzyme is extremely fussy about exactly which E2 enzyme and which protein can interact. The correct E2 enzyme loaded with activated ubiquitin can move and position itself correctly on the E3 ready for action. When both protein and ubiquitin are loaded onto the E3 enzyme, they're brought close enough together for the ubiquitin to be transferred to the target protein substrate either directly from the E2 or through a short hop via the E3. This process can be repeated several times to create a polyubiquitin chain on the protein. The creation of this chain is the death knell for the protein. It provides a clear signal to the cell's waste disposal unit, the proteasome, to start work. The proteasome binds and removes the polyubiquitin chain and unfolds the protein. The protein is threaded through the proteasome chamber, where it's chopped up into building blocks to be reused for the synthesis of new proteins. The ubiquitin can also be recycled. As we've seen, the UPS is a critical process in controlling cell function. When protein degradation gets out of balance, disease results.